Okay, so welcome to this next video in which we are discussing type 2 activation of endothelial cells. Okay, so we've seen now that in type 2 activation of endothelial cells, you activate the activator protein 1 and the nuclear factor kappa B, which are going to increase the expression of all sorts of genes. Now, the only gene that we've seen and looked at so far is this cyclooxygenase 2 enzyme, and this isn't going to do anything new. It's just going to speed up a process that was already there in type 1 activated endothelial cells. So in type 1 activated endothelial cells, what has happened is that histamine or some other ligand has come and activated a G protein coupled receptor that is coupled to the GQ heterotrimeric G protein. This leads to the activation of the alpha Q GTP enzyme, which leads to the production of inositol 1,4,5-trisphosphate, IP3, which then leads to the opening of the IP3 receptors and the release of calcium from the intracellular stores. Okay, this calcium that you've released from the intracellular stores will then bind to a C2 domain of the cellular phospholipase A2 enzyme, and it will then migrate from the cytoplasm to the inner leaflet of the phospholipid bilayer. There it will start breaking down the phosphatidylcholine molecules in the phospholipid bilayer into lysophosphatidylcholine and arachidonic acid. Arachidonic acid will then be converted by this cyclooxygenase 1 enzyme, which is constitutively expressed in the phospholipid bilayer of the endothelial cells, into firstly prostaglandin G2 and then into prostaglandin H2, and prostaglandin H2 will go into the cytoplasm and be converted by the enzyme prostaglandin I2 synthase or prostacyclin synthase, which is in the membrane of the endoplasmic reticulum. Okay, you will then secrete the prostacyclin, and we'll discuss what it does in a moment. However, basically when you have type 2 activation occurring as well, what's going to happen is you're going to produce a new enzyme known as cyclooxygenase 2, and this is just bigger and better than the cyclooxygenase 1 enzyme. Basically, it can convert arachidonic acid into prostaglandin H2 far more rapidly than cyclooxygenase 1. It's much better at doing it. So the output of prostaglandin H2 that you are producing will markedly increase once you've started to express cyclooxygenase 2. That means that the output of prostacyclin will also markedly increase. So basically, type 2 activation of endothelial cells is going to increase the secretion of prostacyclin that had already begun by type 1 activation of those endothelial cells. Okay, right. So what does the prostacyclin do? Well, basically, it's going to be secreted by the endothelial cells, and it's going to cause relaxation of smooth muscle cells. Now, back to our blood vessels, and I told you it would be worth doing this initial work here. So basically, which blood vessel is it going to be important that we are secreting a relaxant molecule? Well, it's going to be the arterioles, basically. The endothelial cells of the arterioles are going to secrete this prostacyclin. It's going to go back to the smooth muscle cells that surround them and cause those smooth muscle cells to relax. Okay, and when they all relax, their lengths will all go up, which means that the circumference of this ring of vascular smooth muscle cells will increase, which means that the diameter of this ring of vascular smooth muscle cells will also increase, which means that the diameter of the lumen will increase. So that's going to mean that you get vasodilatation of these arterioles that supply the affected or infected area. Okay? So you're going to increase blood flow to that affected area, and that is going to further produce the rubor and calor, basically. So type 2 activation of endothelial cells is going to upregulate the blood flow to the area even more than type 1 activation had already done, basically. And that's going to promote rubor, redness, and calor heat. Okay, right. Now, as I say, it's very important that the endothelial cells of the arterioles produce this prostacyclin. But if the endothelial cells of the capillaries and venules produce prostacyclin, you might think, well, what's the point of that? Because they have no smooth muscle cell around them. But you have to remember that they'll be secreting this out into the interstitial fluid around them, basically. And, you know, as I said before, there's not just going to be one arteriole with a capillary bed leading to a venule in this area. There will be multiple arterioles, and maybe there's another arteriole that just happens to be near these capillaries and venules, okay? And now the prostacyclin that they are secreting may well make its way over there, and it may well cause the vasodilatation of that arteriole, which is also supplying this affected area. So it's not as though the production of this prostacyclin by the capillary and venule endothelial cells will be utterly wasteful. Okay, right. So, that's cyclooxygenase 2 now discussed. Let's now go on to another effect of type 2 activation. Okay, and now this is where things are a bit mysterious. They're not known. Okay, so I'm not actually going to be able to tell you what proteins we're actually upregulating. Okay, so what's going to happen is that our two transcription factors, nuclear factor kappa B, okay, NF kappa B, and activator protein 1, these are both going to activate the expression, or upregulate the expression, of what I can only call unknown effector proteins. Okay, so we do not know what these proteins are. Okay, but we know that some protein is created due to NF kappa B and activator protein 1. Okay, so unknown effector proteins. Right, so what are these unknown effector proteins going to do? Well, basically, they are going to reorganize the actin and tubulin cytoskeleton. Okay, so what do I mean by this? Well, let me draw a picture. So you remember me telling you when we were discussing type 1 activation of endothelial cells that the type 1 activation triggered the endothelial contraction. It triggered the endothelial cell to get a little bit narrower, okay, and pull away from its neighbour over here. So here is its neighbouring endothelial cell. Okay, well basically type 2 activation is going to also do this. But it's going to do it maybe to a bigger extent than type 1 activation. And it's going to actually lead to a complete reorganization of the actin and tubulin cytoskeleton. So it's not just going to pull, it's not just going to contract this away, it's actually going to change the cell's structure so that it no longer exists over here, and it's going to actually move its boundary backwards. So it's not just a contraction, it's a actual permanent change. Right, so basically what you're going to produce is you're going to produce unknown effector proteins that are going to interact with two components of the cytoskeleton, so actin and also tubulin. So these are both little proteins which polymerize to make huge, great strands which are involved in producing the cytoskeleton. And just in case you're not really comfortable with what a cytoskeleton is, let me give you the motivation for what a cytoskeleton is. So basically, you think of cells having a rigid structure. But the question is, why do they have a rigid structure? Is it the cell membrane that keeps them with a rigid structure? And of course the answer is no. The cell membrane is just a gooey mush of phospholipid. It would have no structure, it would just be wobbling around like a jelly. So it wouldn't give you a solid structure. In fact, a jelly does generally have a reasonably consistent structure. So I don't know, something even more wobbly. Just imagine some sort of computer graphic that's oscillating continually. Okay, it would be like that. Right, but basically what you do have is a consistent cell structure, so you must have some sort of rigid structure. So basically inside of the cytoplasm of the cell you have an incredibly dense meshwork. Now we never draw this when we're drawing pictures of cells because it would make our picture absolutely hideous, because what we'd have to draw is something like this. We draw just fibre after fibre after fibre after fibre, and you see how hideous this is. Basically, you've just got protein everywhere, making up a very rigid and intense and dense structure that is just holding the cell membrane up and keeping it in that shape. So basically, you have protein filaments everywhere in the cell, and two of the major proteins that are used as cytoskeletal components are actin and tubulin. Okay, so these are little proteins which can be polymerized together to make actin filaments and tubulin filaments. Okay, so what's going to happen is that when the cell undergoes type 2 activation, the nuclear factor kappa B and the activator protein 1 are going to increase the expression of proteins that are still not known, which are going to cause the retraction of the cytoskeleton over here, basically at the poles of the cell. So what's going to happen is, if you can imagine, there was a cytoskeleton, so let's say we're going to retract ourselves to here. So there was cytoskeleton in this terminal little bit here, so I'll colour in it so I have something to point to. So this turquoise bit that we're going to take away, basically, there was cytoskeletal components in there. There was a thick meshwork of actin and tubulin. If you want to dismantle that portion, what you need to do is remove the actin and tubulin, basically. And that's what the cell will do. It will start to remove those actin and tubulin proteins from there, and basically the cell membrane will just flop once you've removed the support that was holding it up. So that's how you're going to retract these two cells. So you'll do it to this one as well. And you can see that if you retract and remove these two turquoise pieces now, you are going to end up with a much bigger gap between endothelial cells. Okay, so basically in type 1 activation of endothelial cells, you've got a quick fix. Okay, you've got endothelial contraction, where basically the phosphorylation of the myosin light chain led to the contraction of actin filaments, which just sort of pulled the end of the endothelial cell this way. So it wasn't actually a permanent change. If you'd stopped the contraction process, it would have sprung back, basically. Whereas now what we're going to do is actually remove the cytoskeleton from there, and we're making a permanent change to the structure of our cells, which is going to lead to a permanently bigger, well, not obviously you can reverse it, but it's a more permanent whole than the type 1 activation had produced. So basically, what this is going to achieve is it's going to make the capillaries and the venules in particular, rather than the arterioles now, it's going to make the capillaries and the venules in our area more leaky. Okay, so if we get our picture of the capillaries and the venules back, okay, if I can just find it, here it is. So here we have our capillary, and here we have our venule. If you can imagine opening up all these gaps in between the two cells, then we're going to now get holes in the side of our venules and in the side of our capillaries. What this is going to allow is it's going to allow fluid and protein, but these holes aren't going to be big enough, not quite big enough for cells just to move through, not without help, and we'll discuss help in a moment. Okay, but basically, fluid and proteins that are within the blood are now going to move out of these holes and into the interstitial space. So what you will form is an inflammatory exudate. So you'd already done this from type 1 activation, but now the holes are bigger, basically. So you're going to form even more inflammatory exudate, basically. So the amount of inflammatory exudate you have formed will go up even further. And you're going to bring in even more proteins. You'll bring in even more complement proteins, and again, you'll bring in more coagulation factors. And the reason that you get far more of these now is because previously, when we just had type 1 activation, the holes between the endothelial cells that type 1 activation was capable of producing were not great, basically. They were a little small, and these proteins, these complement proteins, and in particular some of these coagulation factors, they are big proteins, so they were struggling to get through those holes. So not many complement proteins and coagulation factors came through the holes when we'd just undergone type 1 activation. Once we've now undergone type 2 activation, we have much bigger holes in the side of our capillaries and venules. So you're going to get complement proteins and coagulation factors pouring out, basically. And this means that the complement factors can now attack the Staphylococcus aureus bacteria directly, so they're involved in cytotoxicity, and the coagulation factors can now undergo the coagulation cascades, both the intrinsic and extrinsic coagulation cascade, which they will do just automatically. As soon as the coagulation factors leave the bloodstream, they undergo the coagulation cascade, and you'll get fibrinogen being converted to fibrin, okay, and that will mean that you produce a very dense meshwork of fibrin strands that will sort of ensnare the Staphylococcus aureus bacteria. So imagine a very thick spider's web that surrounds each and every Staphylococcus aureus bacterium and just prevents them from moving anywhere, basically, and prevents them from dividing because they simply don't have the space to expand into. Okay, so it's going to contain the infection. So bringing in these proteins are very, very important in the innate immune response. Okay, right, so there's another part of type 2 activation of endothelial cells. We're going to increase the expression of these unknown effector proteins, which are going to cause the retraction of the edges of the endothelial cells so that you increase the size of these holes between the two endothelial cells. So again, this is another example of how we're just improving on something that had already occurred in type 1 activation. So both of the examples we've seen so far are just improvements to type 1 activation. COX-2 was just an improvement of COX-1, and this is just an improvement of the endothelial contraction that you got in type 1 activation of endothelial cells. In the next video, what we'll do is continue this discussion.